Let my little tidbit there discourage you. If you have the chance to take developmental biology, do it. If you want to. I'm not your boss. Okay? Cool. Let's do this. Basic embryology. Embryology is the study of the origin and development of a single individual. What does that mean? It's the study of how we form. Okay? There is one period that we kind of talk about when we're talking about development. It's the prenatal period. Prenatal just means before birth. So the before birth period. And that period is broken up into two stages. The first is the embryonic stage. And then the second is the fetal period. Okay? So much happens during that first 8 weeks, your embryonic period. Lots of things go down. And we're going to talk about them. Okay? So when we're looking at these stages, the first 8 weeks, the embryonic period, we start with fertilization. That is the coming together of that sperm and the egg. That happens, okay? About a week later, you have some cells growing. We'll talk about all those cells and all of their many different names and the names that they change as they kind of get a little bit bigger and a little bit bigger and a little bit bigger. Eventually, at around 3 weeks, you have an embryo. That embryo is about 3 millimeters in size, so it's super tiny. I can't even, 3 millimeters is maybe half of my pinky nail, okay? So tiny. About 5 weeks, the embryo is about 10 millimeters, it's a little bit bigger, it's growing some stuff. I think it looks like an alien. I call it the alien baby phase. You'll hear all of my weird phases that babies, they're great, we love babies. Babies are cool. Don't do that. And then about 8 weeks, you have some other things developing, some major organs, the basic body. And then the second half, it's not really half, but the other part of the prenatal period, the fetal period, is when you're straight up a fetus. That's 12 weeks until you're born, until natalness. I don't think natalness is a word, but you know what I mean, until you're born, okay? And during that time, your organs grow in complexity. You develop fat, you develop all kinds of stuff. Eyeballs, skin, hair, you practice breathing, all kinds of things happen. Okay? It's kind of that basic body plan. If you remember from chapter 1, we kind of talked about how we are basically a tube in a tube, right? This is where that tube in a tube kind of starts to come into play. So your skin is your dermis and epidermis. That's kind of the outside of the tube, and this will make more sense with the pictures as well. The outer body wall has your trunk, your muscles, your ribs, your vertebrae, all of that stuff. The body cavities, your digestive tube, that's the inner tube. That's the tube that's within the tube. You with me? And then your limbs. Your limbs will eventually sprout off of the sides of the trunk of the little tadpole thing that becomes a person eventually. Okay? This is, don't let this image scare you. If you cut me in half just like this, you'd be able to see one layer out here, that's that ectoderm. That ectoderm has your skin, your dermis, all that, and we'll get into what all of these layers are and what they contain. The middle layer, your mesoderm, and then the inner layer, your endoderm. Let's break down those words a little bit. Ecto, what does that mean? Outside, yeah. Ecto, like ectoplasm, ectopic pregnancy. Ecto is basically just outside, or out of, I think. Mesoderm, what is mes? We kind of talked about it a little bit with metaphase. Middle, yeah. The mesoderm is the middle layer. And then the endo That term endocardium, endoplasmic reticulum, endo, inside or within. Okay? So that's kind of the first step of the breakdown. Embryology can get quite complicated. Don't let it scare you. Okay? You with me so far? Great. So, the embryonic period, the very first week after you have joined the egg and the sperm, that is the joined egg and sperm is what we call a zygote. Okay? The zygote, the fertilized egg, actually gets fertilized in the fallopian tube. And then using those, what are these called? Cilia, it gets pushed into the uterus eventually, where it will then implant. Okay? The zygote starts as technically 2 cells that became 1, and then that 1 cell divides into 2, and then into 4, and then into 8, until it, 8, until it starts developing exponentially. And then you'll have a whole bunch of cells. And that cluster of cells will eventually become something. Okay? So cleavage, that's that kind of separation of the zygote from itself into a secondary cell. That's what we call cleavage. At the 12 to 16 cell phase, the collection of cells is called a morula. Okay? That morula will continue dividing and become this fluid-filled structure called a blastocyst. Blastocyst has some fluid and about 64 more cells here or there. Okay. That blastocyst, we kind of talk about it in 2 parts. We have the inner cell mass, which will eventually form the embryo. And then we have the trophoblast, which helps to form the placenta. That was a lot of definitions. Okay. So let's break it down a little further. In the very first week of development, the zygote is just 1 sperm, 1 egg, which should equal 2, but it doesn't. It equals 1 zygote. You with me? 1 plus 1, in this case, equals 1. Okay? Eventually, that zygote is going to divide. Then you'll have, another cell that is here. And those cells will also divide. And then you'll have 4 cells. And that is the 4-cell stage. It hangs out as 4 cells for a couple of, up to a day, I think I want to say. Okay? After that 4-cell stage, we, guess what, continue dividing. And eventually we will get to a morula. Morula has about 12 to 16 cells. Ish, somewhere around there. That morula will then implant to the uterus and be called a blastocyst. But later, late blastocyst. Early blastocyst is still just kind of hanging out. About 64 cells, give or take. And it has 2 components to it. It has the inner cell mass and the trophoblast and a bunch of fluid. H2O. Okay. And then the very last bit is the late blastocyst stage, and that's where we get uterine implantation. All of this is within 1 week. This happens quick. Okay? Don't let this image scare you. This is just kind of showing the path that that egg will take throughout the first few days of development. So as you ovulate, that egg starts to move through the fallopian tube. Eventually, a sperm will maybe hit it, find it, and they will join. That 1 + 1 becomes 1. As we move through, at around day 2, you'll hang out as 4 cells. Around day 3, you're a solid little ball clump of cells. Around day 4, that morula starts to hollow out and become the early blastocyst with the inner cell mass and the trophoblast and the fluid. And then a little around day 7 is usually where you'll see some implantation. So it hangs out as that early blastocyst for a little bit. Okay? Okay. So I talked about that inner cell mass, right? That is one of the parts of our blastocyst. Okay, that inner cell mass is what we call a bilaminar embryonic disc. Embryonic disc. Okay, that bilaminar embryonic disc is the inner cell mass divided into 2 parts. Those parts are the epiblast and the hypoblast. So 1 epiblast plus 1 hypoblast equals bilaminar embryonic disc. Okay? The amniotic sac is formed by an extension of that epiblast. The embryonic sac is basically just that water balloon that fetus will hang out in for the next 9 months. Okay? That amniotic sac has a bunch of fluid in it called amniotic fluid, which will eventually, when the sac breaks and you're ready to start giving birth to that baby, it pops, breaks, and says, oh no, go to the hospital now. Now, run. Okay? Maybe run, maybe not. Depends on if you're lucky. Okay? The outer membrane of that epiblast forms the amnion. The inner membrane forms the amniotic sac cavity. The amnion is just kind of the layer on the outside. Okay. That bilaminar embryonic disc has 2 parts. We talked about what the epiblast forms into. Let's talk about what the hypoblast forms into. The hypoblast forms into the yolk sac. The yolk sac will eventually become your digestive tract or your digestive tube, right? Remember, we're in a tube within a tube kind of thing, okay? The yolk sac, unlike in a chicken, is not a major source of nutrients for the embryo. The embryo gets its nutrients from the placenta, which is formed from the trophoblast. Talk about that later. Blastocyst phase. Okay? The tissues around the yolk sac will eventually give rise to your very early blood cells and blood vessels, but for the most part, those form in later in the mesoderm. Cool? Cool. All right. So this is just kind of showing that blastocyst pre-implantation. I don't know if y'all can see it, but there's a tiny little gap in between that blastocyst and the wall of the uterus. It has not quite implanted just yet. Okay? Day 6, it starts to adhere, and you get that separation of the inner cell mass and the other parts. Implantation proper. Then you get the trophoblast starting to invade into the uterine wall. Again, this trophoblast will eventually become placenta. Later on, day 9, you have that broken up inner cell mass made of hypoblast and epiblast. Right? And eventually, it starts to look a little bit more complicated. You got your bilaminar embryonic disc hanging out right in here. I know it's kind of hard for you guys to see that, but it's right in there. Too far. Right? Epiblast, hypoblast still there, just growing, dividing. And all throughout this time, these are all cells that are divided. So you have, hell, probably a few hundred, maybe 1,000, maybe less. Don't quote me on that. Cells by the time you're at like day 11 blastocyst. Okay. So at this point, we are at a place where the cell has the epiblast portion, it has the hypoblast portion, we have our bilaminar disc, that little section in between where the epiblast and the hypoblast meet. Okay. Eventually, we will form what is called a primitive streak. The primitive streak is a raised groove on the dorsal surface of the epiblast. What's the dorsal surface? The back, yeah. So it's on the back half of the epiblast. So probably somewhere around here. Okay. Okay. And then the process of what we call invaginating the epiblast cells, which is basically just kind of like folding itself in over on itself, is called gastrulation. Oh, this is black. Gastrulation. Okay. Gastrulation starts at the primitive streak and then forms 3 primary germ layers. Those germ layers are your endoderm, your mesoderm, and your ectoderm. The endoderm is formed from migrating cells that replace the hypoblast, because the hypoblast is eventually probably going to get away from us a little bit. Okay. The mesoderm is in between the epiblast and the endoderm. And then the ectoderm is formed from the epiblast cells that stay on that dorsal side. Okay? Something to note is that all of these layers are derived from epiblast cells. See that? Great. All of those layers are derived from your epiblast cells. Your epiblast and your hypoblast. Epi, hypo. Okay? Don't forget. So when we're talking about the invagination of those epiblast cells, this is kind of what we're talking about here. So if this blue layer up at the top is the epiblast, and the hypoblast is kind of right down below there, these cells are moving from being in the kind of epiblast region to pushing the hypoblast cells out of the way. Okay? And those secondary cells that move everything out of the way will start to become those primary germ layers. You with me? Kind of. It's complicated, I know. Okay. So one of these structures that forms from that invagination is the notochord. Okay. It starts with the primitive node. The primitive node is kind of a little swelling at the end of the primitive streak. Remember, we had that primitive streak that formed earlier on from the epiblast and hypoblast. And then the notochord, which is formed from that primitive node, eventually defines your body's axis. So telling your body which way is your head, which way is the tail, where should I put the arms, where should I put anything. So that notochord helps to define which way is up and which way is down. It's formed from mesodermal cells and a few endodermal cells. It will eventually be the site of your vertebral column. It shows up at day 16, but it doesn't reach your noggin until about day 18. Okay. So if this is what we were looking at here, our whole guy, the blue ones are still our epiblast cells, right? We got the invagination, so we got those layers, the embryonic disc layers, ectoderm, mesoderm, endoderm. Eventually, that kind of lengthens, starts to break open, but not really open, right? But starts to kind of elongate and becomes those two separate pieces to them, where you have your notochord starting to form kind of in that mesodermal layer, mesodermal-ish layer, and you start to form the other layers as well. Okay. Neurulation is the process of the ectoderm forming your brain and spinal cord. Okay. Remember that we have ectoderm, mesoderm, endoderm. And all of those were formed from our overall epiblast. Does that make sense? So your ectoderm will start to form your brain and spinal cord. That's called neurulation. Your ectoderm in the kind of middle part will form a little plate. It's not exactly a plate, but it's like a little kind of grooved plate there. Eventually that neural plate will fold kind of in. These images, the images that are here are better than what my arms can tell you, but they kind of fold inwards like that. And that's where you form your neural tube. That neural tube will eventually become like where your spinal cord is. The cranial part becomes your brain. In utero, the carrying parent's mineral balance is super important to the proper development of that embryo, right? It's because of certain chemical changes in the mother can affect development down the line. Because all of the induction of cells becoming, because remember, these were just two little cells once upon a time. They're eventually going to become all of your organs, all of your skin, everything that is inside of your body will eventually become something from those one cells. In order for that to happen, we do a process, not we, ourselves, do a process called induction. And that induction is kind of based on the chemicals around around the cell to tell you, hey, because I have this gene, and this chemical next to me, I'm going to become a heart cell. I have gene 4, gene 10, gene 11, and way too much sodium, I'm going to become a kidney, right? So it's that process of how the chemicals in the mother's body help to inform the eventual pathway of the embryo. Okay? Yeah. So the epiblast cells will form your ectoderm, your mesoderm, and your endoderm. And then that ectoderm will eventually form your brain and spine, null cord. Okay. The neural crest also originates from those ectodermal cells, and they will form the sensory nerve cells. Okay. So when you think ectoderm, think your nerves. Your skin a little bit, your skin a lot of it, but mostly your nerves. Your nerves and your skin, ectoderm. And then, like I said, induction is the ability of one group of cells to influence the development of another group of cells. They can do that. You can just bully a bunch of other cells and be like, hey, all y'all, heart cells, because I'm a heart cell. So you're all going to be heart cells with me. Let's go. If you wanted to be a liver cell, you could be like, hey, all y'all, liver cells. Why? Because I said so. Let's go. And they'd all become liver cells. It's more complicated than that, but the gist. Okay? Well, that's not what you want. All right. Okay. So the mesoderm. The mesoderm is kind of broken up into three parts. And those three parts are somites, intermediate mesoderm, and then lateral plate mesoderm. Okay? So we know that the notochord is formed from mesodermal cells. Right next to that notochord, we have the somites, or the paraaxial mesoderm. Okay? Somites, paraaxial mesoderm, they're the same thing. These somites are segmented. And eventually that segmentation will turn into other things throughout the body, but we'll talk about them in a little bit. All right? The intermediate mesoderm is just next to those paraaxial mesoderms. It is also segmented. And then we have the lateral plate mesoderm. These are unsegmented, meaning that they do not break up. They are mainly just sheets of tissue. Okay? Lateral plate mesoderm, like its name states, is the most lateral part of the mesoderm. What does lateral mean? To the side, right? Later means side, right? So it's on the sides part of the mesoderm. That, like I said, remains unsegmented, but it splits into two layers, all right? I'm not going to draw everything else on the sides here. But imagine there's a whole bunch of notochord, intermediate, you with me there, kind of stuff. I'm not going to draw all of that. But the lateral plate mesoderm breaks into two parts. One that kind of wraps around the back, and one that kind of wraps around the front. And those are the splanchnic and the somatic mesoderm. Okay? All right. The splanchnic mesoderm will eventually become, I'm sorry, this is flipped. I'm so sorry. My bad. Showmatic. Splanchnic. I might have mixed those up. Sorry, guys. All right. Okay. And then the space between those two sheets is the Salem or Colium. I never know how to pronounce that word. Who puts an O and a E next to each other? What sound does that make? For my linguists in the classroom. Lord knows I don't know. Okay. This is just kind of going to show you the phases of neurulation kind of in a more succinct way, right? So we have that notochord. The neural plate starts to form. It's that thickening, right? We get thick, then we groove, then we tube. We get thick, then we groove, then we tube. And it eventually pinches off. Okay? So you'll have a layer of ectoderm, a little bit of mesoderm in the middle, and then another layer of ectoderm. Okay? The folding of the embryo is complicated and complex. Essentially, you will go from having the notochord. Imagine that my body is the notochord itself, okay? And then these little bumps that I'm putting on my body, those are the somites that are hanging out next to me on the notochord, okay? The embryo will fold from the back, out, and around. Does that make sense? I know it's weird and it's complicated, but I talk about this with cats a lot, right? So a lot of you probably don't know, but cats, when they develop, you ever see a cat with like colors on the front and then on the bottom it's like just a flat white cat? It's because all the printer ran out of ink. Printer, mom. It's actually genetic. So they develop from the back down, and that's very similar to how we develop from the back kind of down. But instead of going down, we kind of fold this way, right? And have arms and legs as opposed to a cat that has paws. Does that make sense? Okay. So in the same way that a cat develops from back to front and the bottom will be all white and the top will be all like nice and colorful, is kind of the same way that human embryos fold. You with me? Mm-hmm. Okay. All right. And when we have done that fold and eventually formed up, you'll have a little tail. You'll have one end that is the head region, one end that is the tail region, and you end up looking kind of like a little tadpole. This guy, right? So by the end of, let's say, end of week 1, week 2, you are at tadpole formation, right? At around 1 month, that tail starts to disappear. So you did have a tail once upon a time, and unfortunately or unfortunately, it is gone now. But that's why we call it the tail bone, right? Tail bone, tail, cool, okay? The primitive gut is enclosed. Maybe doesn't want me to write primitive gut today. You all see that, right? I tried and it was erasing letters as I was writing them. That wasn't my fault. The primitive gut is what encloses kind of that tubular part of the yolk sac, and that will eventually form your digestive tubes and your respiratory tubes and the respiratory structures, right? Okay. All right. So derivatives of the different germ layers. I talked about it a little bit. The layer that forms your brain, your spinal cord, your epidermis, the most outermost layer. Do we remember what that one's called? What was that? Ectoderm. Remember that root ecto means out. So the ectoderm forms your brain, your spinal cord, your epidermis. The neural crest cells form melanocytes, some of the bones of the face. But remember, those neural crest cells are. Let's not get too confused. What layer forms the internal of your body? Endoderm. The endoderm. Good. And that leaves one layer that is a little more complex than the other ones. It had a bit more parts to it, but it is called the mesoderm. The mesoderm. Good, perfect. Okay. The notochord is a segment of the mesoderm. Okay. Remember that notochord. We go notochord, somite, intermediate, mesoderm, and lateral plate. Mesoderm. Okay? That's why the mesoderm is a little bit more complex. Eventually, that mesoderm will give rise, or part of that mesoderm will give rise to the nucleus pulposa in your intervertebral discs. That is essentially just the core of your intervertebral discs that can handle that compression from walking, moving, jumping, all that stuff that you do that makes your back hurt in a day. There's the stuff in between that makes your back hurt a little bit less. If you didn't have that stuff in between your backs, oh Lord, it wouldn't be a fun time for anybody. Okay? All right. The mesoderm. I told you it's segmented, right? Those segments divide into different things. The somites divide into your your sclerotome. Jesus. Sclerotome. Thank you. Your dermatome and your myotome. All right. Your sclerotome will eventually turn into the vertebrae and ribs, and ribs at the respective site where they end up kind of falling. Okay. Your dermatome will end up forming the dorsal dermis. So not the epidermis, but the layer underneath on the back side of your body, the dorsal dermis. Okay. And then the myotome will eventually form your limb buds and parts of and segmented parts of the trunk muscles. Okay. All right, your intermediate mesoderm forms your kidneys and your gonads. So your genitals, your kidneys, your intermediate mesoderm forms those. And then the lateral plate mesoderm, unsegmented mesoderm is the same thing as the lateral plate mesoderm. Okay. And then that lateral plate mesoderm splits into two parts, right? Remember that? Splanchnic and somatic. The splanchnic mesoderm ends up forming the majority of your musculature, a lot of your connective tissues, some of the linings of your digestive and respiratory structures, as well as the heart and most of your blood vessels. Okay? And the somatic mesoderm ends up forming the dermis on the rest of your body, the bones and ligaments. Okay? Don't let this image scare you. It's just to show you kind of how that body folds its fold. This flowchart, hint, hint, nudge, nudge, wink, wink, important. I would know what this flowchart says. This flowchart is essentially showing you that all of your germ layer cells are derived from the epiblast. Okay? Derived from the epiblast. The ectoderm and what it forms, the mesoderm, what it forms, and the endoderm and what they form. Okay? I would know this stuff. Oh, actually, all of it, the whole chart. It's a good chart to know. I would know this chart. Okay? Cool. And that, guys, finishes up the majority of the embryonic phase. Weeks 5 to 8 is just kind of where a little stuff starts to develop a little bit more. You start to form limb buds. The embryo starts to look a little human. I don't think so. I think it's an alien still, kind of. I think it looks a little alien-like. Its head is huge. Its body is teeny tiny. I mean, respectively for how big it is. Its head is gigantic. Its body is teeny tiny. But all of the kind of rudimentary early forms of the organs are in place by the end of month 2, which is around 8 weeks. Okay? Hi. Okay. So, right around a month or 2, you look kind of like a deranged tadpole. This is the tadpole stage, about 1 month in. You still have a tail, you still have kind of all that stuff going on, all right? Now let's talk about the fetal period. So you remember that the embryonic period is weeks conception to week 8. Then the fetal period is weeks 9 until you're born. All right? So during the first kind of half of the fetal period, your cells are doing a lot of differentiating. They differentiate through a process called induction. We talked about induction a little bit, basically saying, I'm gonna be a heart cell, so all of you other guys around me, come on, we're gonna go be heart cells together. Okay, that's induction. That induction is initiated by different genes and chemicals in the mother, right? So there are different genes that will turn on some stuff that will make some cells stomach cells or liver cells or heart cells or whatever they are or will need to be. Okay. A normal birth occurs about 38 weeks after conception, but that's an average. Anywhere from 38 to 42 is relatively normal. I was 42. I hung out for an extra 2 weeks and made my poor mother miserable. Okay? Any being that is born before 38 weeks, we would deem premature. Right? Are you gonna let me play the video? I don't think it's going to let me play the video, but watch that video. It's cool. It's an ultrasound at around 20 weeks. All right. So let's talk these a little bit more specific phases. Like we know, anytime before 8 weeks, classified as an embryo. At around the 2-month phase, I like to call this the alien 1 phase. During that alien 1 phase, the head is basically as large as the body itself. It has eyes and ears and like a nose and stuff like that. But I mean, look at it, right? Cool. But the major organs are kind of in place. They're not fully formed, but they're right around where they're supposed to be. Month 3, I call this alien phase 2. Alien phase 2. You have alien phase 1, alien phase 2. Alien phase 2, the brain still gets a little bit bigger. It's continuing to grow. The fetus begins to move a little bit, but not anything that's perceptible by the mother. The intestines start to form, limb buds start to develop a little bit more, all that jazz. All right? Around month 4, we start to develop a little bit more skin. The torso gets a little bit bigger, bones start to kind of form. But remember, those bones don't become actual bones until like after you're born. They're mostly just cartilage at this point that will eventually turn into bone via ossification. Month 4 is what I like to call the raw chicken baby phase. Raw chicken phase 1. Okay? Raw chicken phase 1 is about month 4. It kind of looks like a baby, but a really, really small baby that looks like it had its feathers plucked. Raw chicken phase 1. Okay? Around month 5, the fetus starts to get a little bit bigger, a little bit longer, not necessarily any like chunkier, but a little longer. So that's what I like to deem raw chicken phase 2. Right? Around month 6 and 7, you start to gain a lot more weight. Still doesn't quite look, at least to me, like a full human baby. I like to call this the skinty baby phase. Maybe. Skinty baby phase. This is where the kind of distal limbs, so like the farther part of your limbs, begin to start to ossify. They start to kind of form what could potentially be bone later on. Your cerebrum and cerebellum start to continue to grow, and this is where the majority of your brain growth happens. You get a lot of brain growth happening in months 6, 7, 8, 9. Okay? The lungs finish developing right around week 28 or so, and that is approximately when you could theoretically survive outside of your mother. Okay? And then month 8 and 9, this is our chunky, sweet baby phase. Chunky baby phase. And that's basically where you're just adding and accumulating a lot of fat and getting all cute so that when you pop out, you don't look like a raw chicken entirely. You still look kind of raw chicken-like because, you know, babies are small, but it at least looks like a baby. Okay? Does that kind of make sense? Mm-hmm. So in your book, it will describe a lot more of what goes on in each week, each month of a pregnancy. So read through those to get kind of a better idea of what happens month to month to month. This is a pretty good like overview, but if you want details, textbook. All right? So the phases as I deem them, and that's just me, you know who I am.